Here we are at Rothamsted Research in the UK, a plant research institute that's not only home to the longest continuously running agronomic experiments in the world, but also a leader in cutting-edge, modern plant genetic engineering research. Today we're going to study the process and the value of the scientific method by looking through the lens of one particular case, the Whiffy wheat case. Let's take a look at how science works, how scientists identify questions, formulate hypotheses, test those hypotheses, and then, based on the data that comes in, refine those hypotheses. We'll also look at the importance of transparency and openness at institutions like Rothamsted and what that means for public trust and understanding of how science works. Let's go in. Today we're going to talk to 4 scientists who worked on the Whiffy wheat project. The project leader, prominent chemical ecologist John Pickett, Hugh Jones, the research leader of the Cereal Transformation Group, chemical ecologist Gia Aradottir, and Toby Bruce, a research scientist studying plant-insect interactions. So Professor Pickett, can you tell us a little bit about the Whiffy wheat project and what questions you're really trying to answer and how you got involved in this
Well,
work?
I'd certainly love to tell you about it. We've been thinking about this project since the early 80s, in fact. I published a paper in 1984 saying that one day we would be able to use genetic modification of plants to produce crops with chemicals in them. And one of the chemicals I chose was the aphid alarm pheromone. This is a pheromone that aphids, small insect pests, use to warn of danger, and so when they smell it, they scuttle off the plant. These aphids carry viruses to plants, and so we have to control them, and we do so by using a number of fairly broad spectrum eradicant pesticides. These are chemicals that actually are designed to kill aphids and not us. They're very good at it, but we usually see resistance developing, and so we feel that some of these softer approaches, like the aphid alarm pheromone, could replace insecticides for the most part.
Fundamentally, aphids are pests of wheat, and what they do is they suck the sap out of wheat, and they can also transmit viruses, just like mosquitoes. Farmers can use up to 40% of their yield because of this virus. So traditionally, aphids are controlled by insecticides, but a better way, I think, would be to get the plant to defend itself against the insects.
And what was your hypothesis in this project?
So aphids communicate via smell. They've got a very strong sense of smell, and they produce an alarm pheromone to warn others of danger in their environment. And when one aphid emits this alarm pheromone, this alarm signal, the others recognize it and they go away from the area. They drop off the plant, they go away, they fly away.
And so the hypothesis was, can we make a wheat plant make this smell? And secondly, if we can, will these plants repel aphids?
So you generate a hypothesis and then you put together a series of experiments to test it, basically. So what we did was, our hypothesis was that if we took a gene that synthesized a protein that would make the aphid alarm pheromone, we could put it into wheat.
So you're the man who makes all of the GM wheat here at Rothamsted.
Yeah, so we take ideas and turn those ideas into gene cassettes, into a test tube essentially that contains a gene, and then we can add that gene to a wheat nucleus, and that gene gets incorporated into the wheat cell. So we've come into the room that we put our little cultures into after they've been shot, so the DNA has gone into the cells, and we now need to turn those cells into whole wheat plants again. And we culture those cells in plates that have special kind of food in them, sugars and salts and plant hormones. And the first thing that happens is that those cells start to grow and form these little lumps, these little little white lumps that don't look much like wheat plants, but they are wheat cells. We can cause some of these wheat cells to start making tiny little leaves. Another few weeks in culture, you can start to see what looks like long, thin leaf structures. Another step after that, we've got what looks much more like a wheat plant now. It's starting to grow erect, and it's got several shoots, and if we then transfer this into soil, it'll form a normal wheat plant. That gene gets incorporated into the wheat cell, into the other 90-odd thousand genes that a wheat plant has. It's now got 90-odd thousand and one, the one that we've added.
So we analyse the plant, we analyse if it's in the plant, and we analyse the air above the plant, because that's where the aphid is going to be. So we have to capture volatile chemicals as they come out of the plant. So we make sure that it produces the chemical and that the aphid can detect the chemical, and we then see if the aphid responds to the chemical.
And what was the aphid's response?
It responded very negatively. It didn't like it. It moved away from where we had the wheat-derived alarm pheromone.
The ultimate aim would be to have these crops grown in the field. We wanted to test the idea that that effect still occurred in the field. So we applied for a license to carry out a field experiment.
The field environment is very different from the lab. In the lab, you have everything is controlled, the temperature is controlled, the light is controlled, the humidity is controlled. But then when you go out in the field, you've got the wind, you've got the rain, you've got everything else, all of the other insects, birds, everything else that's in the environment. And that's something that we have to do in all of our projects. You test something in the lab and then you take it out to the field and you see if it works on a larger scale.
So we were now set, really, to do the ultimate test, which is to go into the field.